Tim McPherson. I'm giving my presentation on the ribosomal biosynthesis of moroidin peptides. This is just conducted as a literature review. I didn't actually do any of this research myself, but moroidin here is a plant product produced by Dendrocnide rhodus, or the Jimpy Jimpy. This plant has been reported as one of the most dangerous and painful plants to touch in the world. This is due to the moroidin product that it produces. It uses it as a defense mechanism to defend itself. And the moroidin has been determined as the base phytotoxin that the plant secretes for its defense. The plant in the middle here is the Celosia argentea. It produces both moroidin as well as Coelogenthin C here. And it uses it in a very similar way as a defense mechanism. As you can see, the structures of both of these are very similar. The only really big difference to the Coelogenthin C is this additional arginine chain on the right side here. And then as well, the moroidin can also just take different structures based on this R group on the side. It can either have just a lone hydrogen, or it can have a OH group where that R group is. The main significance of these peptides is, as well as the defense mechanism of the plant, both moroidin and Coelogenthin C have been shown to have an inhibitory action towards tubulin polymerization. The inhibitory action prevents the creation of microtubules here, that are used in mitosis that help the chromosomes pull apart. And without these microtubules, the cells cannot divide and will eventually die, at least in theory. This gives the possibility for an anti-cancer drug to be produced from these plant-based peptides. The main issue with using these plants to create like a commercial drug for cancers is that they don't yield a lot per plant. Like you just can't get much from them. This is what's creating the interest by researchers to synthesize these actual, like the moroidin and the Coelogenthin C products, so that they can kind of bridge the gap between like a theoretical use and a practical use as a drug. This is stephanotic acid. It was used as the starting structure for the biosynthesis of both the moroidin and the Coelogenthin C. Within the research, it is very similar to the N-terminal ring. As you can see, this is the moroidin, this is the stephanotic acid. Stephanotic acid has been characterized as a ribosomally synthesized post-transcriptionally modified peptide. The peptide is cyclized via a leucine tryptophan residue by a cyclase enzyme from the BERT domain within the genes of the plant. And being able to synthesize the stephanotic acid gave the researchers insight into a potential synthesis pathway for the moroidin and the Coelogenthin C by use of the BERT domain cyclase and the gene transcriptome lining. The actual BERT domain shown here encodes for the plant-specific cyclase that cyclizes the product. The use of the BERT domain cyclase for the stephanotic acid production again gave the researchers the idea to search for similar BERT domain precursors within the moroidin genes to find a similar cyclase, but that can produce a bicyclic product instead, like the moroidin or the Coelogenthin C. Figure A here shows the precursors as well as the entire BERT domain. The precursors are labeled in red and blue, and then as well in purple in a couple slides. And the underlying portion is the actual domain that encodes for the cyclase. The general form that the precursors take is QLLBWR, and then it'd be a blank here, and then H, as the blank can be swapped for a few different amino acids to give different structures. Figures B and C here show different like moroidin chemotypes produced by plant extracts that they tested. Chemotype just describes the subspecies of the plant that have similar chemical structures of their essential oils, but produce different amounts or structures. The actual graphs here are liquid chromatography mass spectrums that show abundance of moroidin-like peptides that are produced by each of the different plant extracts. The Curia japonica, Celosia argentine flower, and the Acacia herb that was expressed via tobacco plant. As you can see, the argentine flower produced the most similar product to moroidin, almost being 100% relative ion abundance. Essentially, it basically produces the exact same mass to charge ratio that moroidin has, which is shown up here, and then over here is the slightly different structure of moroidin. Yeah, so figure C shows it just from the japonica leaf and the Acacia herb extract of the tobacco plant, and the difference between the mass to charge ratios is just the differing structures. One is the extra hydrogen, and one has it's missing that hydrogen entirely. And this one is it fills the alanine as the blank instead of the glycine here, as you can see. Here in figure D, this is again the birth domain and the precursor. It fills the blank with a serine as opposed to a glycine, and it shows similarities from the B. tomentosa seed as well as again the Acacia herb domain extract. Yeah, so the researchers chose to use the cyclase from the Carreria japonica plant here, and they induced the precursor again in the tobacco plant, and they did this to both confirm the ribosomal origins of moroidin, similar to the stephanotic acid and the way it was produced. And the general idea of what the researchers were hoping to do follows along this image. Starting with the top left, this is what was produced by the tobacco plant that had the birth domain induced, and they were hoping to cyclize it here in the yellow areas to produce the moroidin. It's obviously not that simple, but they had to do a lot more because as you see these small jagged edges on the end, it's just cutting off parts of the extract that isn't relevant to the research. So they're just hoping to actually cleave off those parts later, which is here. Yeah, so this is a more accurate representation of what they did. Again, starting with the cyclization where they connected the two bonds there. And then having an endopeptidase cleave off this blue region as well as all the stuff that isn't shown along the left. And then went through a pyroglutamate formation to cyclize this ring from here. And then the exopeptidase cleaves off this orange part as well as everything else that isn't shown there. And the exopeptidase leaves a OH group, leaving a carboxylic acid chain there instead of all that junk that we don't care about. So after they were able to actually produce the moroidin that they wanted, they were hoping to just mess with stuff because they're scientists and they like to do that. So the moroidin was produced via this precursor, and first they just started by swapping each position individually with an alanine just to see if it would still produce the bicyclization. As you can see, some of them did and some of them didn't. It's not really relevant because they don't really use these moroidins for anything as far as I know, at least not yet. In terms of the actual table, these are all the residue areas. So residue 1 through residue 9, all with amino acids in them. Obviously some are blank, but we'll get to that. Yeah, so after they just tried replacing them all with an alanine, they also wanted to try and shorten or lengthen each ring, shown in the green. First, to shorten it, they just removed the second lysine here and didn't produce a bicyclization, so it wasn't really helpful. Same thing, they added a glycine here to lengthen it, also didn't produce the bicyclization that they wanted. However, by removing the blank or the R7 glycine, they were able to produce coelogentin A, which I don't really talk about in this, but more interestingly, on the R6 and R7 down here, by replacing the arginine with a proline and the glycine with an arginine, they were able to actually produce the coelogentin C that they wanted. And yeah, that just produced that. In terms of, again, in the beginning I was talking about cancer cells. So both the moroidin and coelogentin C, after they've been produced, have shown inhibitory action towards tubulin polymerization. The researchers tested both of these products against an array of cancers. Unfortunately, based on tests, only the coelogentin C actually inhibited any sort of cancer, and it only inhibited one in the lung adenocarcinoma, is the one that it actually affected. And it's for some reason, it's the second ring of the structure that's really important to that. I'm assuming it has to do with the arginine side chain in some way. But again, the moroidin unfortunately didn't actually affect any of them. So that's a shame. And in conclusion, mining plant genomic information for the birth domain precursors can help discover other macrocyclic plant RIPs, or the ribosomally synthesized trans-modified things, can help for potential medical impacts. The development of other cyclic peptide-based medicines could be performed through the same searching and screening of like moroidin peptide libraries, as well as specific cytotoxicity of coelogentin C was theorized to be, it could be improved through changes to the core structure, and it could just increase it towards even more types of cancers, or make it better at affecting the one it already does. So, and then that's just the source for the article. And any questions?
Any questions for Tim? I have a question. So the cytogentin C, right? That one was showed toxicity towards cancer cells. So based on your reading, what's like the timeline for that to be a potential like useful cancer drug?
So yeah, so this was just like a laboratory setting. They weren't able to produce a lot of it, from what I understand. So it could be a while. They didn't really, as far as I can tell, they didn't speculate on how long it might take for it to become like an actual useful drug in terms of inhibiting cancers.
Did they compare these two with paclitaxel, which also inhibits microtubule polymerization as a cancer therapy?
Yeah, not that I read in the article, unfortunately, so I could have missed it maybe, but I didn't read anything about that. No, sorry.
Any other questions?